Good morning, everybody, and thank you for being here. Sorry for the long title. I ran out of space. Um, so my topic really, I'm talking about a shallow burial with carbon, and some of the audience in the room here is probably more familiar than the usual audience, but I'll present some of the work we've done here, looking into types of carbon that we use to line the burial pits, as well as the way the carcass is prepped. It's kind of okay. Um, This is sort of a visual for the unfamiliar. The shallow burial is essentially a method of disposal for dead stock, primarily for emergency mass mortality or catastrophic mortality, terminology-wise. This is basically, it's a lined pit. The animal is placed on top of a layer of carbon, a foot of carbon. Soil is replaced and typically seeded on top to essentially dispose of the animals. And it is uh, a more rapid compared to the convention standard of composting the dead animal or the mortality. The practice, again, I can't attach a specific date of its adoption or uptake, but generally after 2015, 2016, the practice started to pick up, and a lot of impressive work is being done around the country on that practice. So really, I'm going to talk about the why, and so uh, yeah, it is not working. I think it, yeah, okay. So So really, the reasons for it is basically like you've seen in the pictures. It completely encapsulates the animal, especially for cases with disease response. This is a safe way to dispose or to eliminate the risk of contagion or spreading. Some of the viruses are actually deactivated. Again, some of the work that is being done around the country tracking the pox virus for swine or African swine fever has shown some deactivation of these viruses. We're also seeing especially from an emergency perspective, the ability to respond quickly is a imperative. So compared to composting, staging, and mixing, it is a relatively rapid process and require less resources, primarily the carbon. And when we're talking about the composting, it's a carbon intensive, especially for a large amount of mortality that has to be managed. But then there are some knowledge gaps, or we can call them concerns from the degradation or the breakdown of the carcass. It's relatively slower than when we talk composting, simply because essentially it is air sealed or encapsulated. So there is anaerobic breakdown, but it's much slower. The other concern is spore forming, or the fact that the risk hasn't been completely eliminated. It's just changing forms or present there for opportunities to arise. Nutrient movement or leaching in particular is one of the bigger concerns, depending on the loading rate, the amount of carcasses that are put in per unit area. And I understand that we have a speaker will touch on that a little more in detail. And we understand also, and the work that I'll present to you will show some of that, is the soil and the site have great impacts on how successful or fast or slow the process will go. So really, this is the kind of big picture. For our research, we really started to look into, especially in North Carolina, when we very heavy swine production, high concentration, when or if we need to resort to using shallow burial with carbon, the carbon resources we have at our disposal is limited. The good quality carbon, more absorbent carbons will probably get exhausted much quicker, and then we'll have to resort to different types. The question we're trying to answer, what is the impact of different types of carbon on that process? And I'll tell you how are we measuring the shallow burial with carbon, or I'm using the acronyms SBS. The other practice that we've looked into is actually macerating or grinding that carcass before burial. So not just whole, but burying it ground versus whole, and getting to see if there's a difference there as well. So I'll take us quickly to the work. So the work was done here in the research station, a horticulture research station in Sampson County, eastern part of the state, one of the top states, top counties in the state for hog production. It's actually the second county in terms of hog production in the entire U.S. So we picked the horticulture research station. It's administered by the Department of Ag in the state. And what I'm pulling here is the soil survey report for the site or the plot where we did the work. And it's a sandy loam or loamy sand soils. And what you're seeing on the right-hand side is the field superimposed with the classification from the soil survey tool. actually looking specifically on how suitable is the site for shallow burial with carbon, because that's an added resource on the soil survey that you can actually use it to rank sites. So keep that in mind. We are looking at 4 types of carbon. So we looked into what possibly we can have at our disposal or available to us. So hay is widely available on hog operations in North Carolina, and we, especially coastal Bermuda and the fescue. We also looked into wheat straw. We have wheat crops. The availability of wheat straw is variable. Corn stover was actually quite difficult. After we committed to using corn stover, we do not bale and we do not have corn stover, which is strange for our Midwestern neighbors. And some hardwood mulch. So these really were the 4 carbons that we proceeded to use. And we, from an experimental design, we looked into sort of blocking by time. So the whole approach was we'll bury these carcasses and we'll dig them at different times. 3 months after, 6 months, and then 12 months, and track the degradation. Those are the coding for the variables. We looked at 4 carbons, 2 states of the carcass, and that very colorful table on the right is actually the individual burial plots, representing 72 burial plots, 24 by 3 timestamps. So 3 replicates per each timestamp. This is some of the pictures of the prep for the site. First challenge is, well, a foot is a unit of volume. Each 4 carbons have completely different densities. So if you're trying to pack a foot of material, how much grams or weight, which the absorbance is mass dependent, not just volume. Especially we saw that with hay as a big challenge. On the right, apologies, but I mean, we are in a mortality session. So on the right-hand side is after the animal is placed, or the carcass is placed on top of the carbon. And the yellow ribbon, again, rookie mistake, comfortable sharing that here. These are data loggers. Easy, very lower complexity, and they record and they store the data. Should have checked if they are magnetic. Could be found with but we managed to recover them, but that was a treasure hunt. But the good thing is we're getting the temperature of exactly what we are tracking for. These are hobos, loggers. This is the carcass grinding. Many of you probably are familiar with the kind of top-loaded kind of TMR mixer, basically, for using macerating the carcasses, and it basically looks something like this. So essentially, after mixing, the elevator dumps the blend of carbon and the carcass for staging or putting in the burial plots. So what we measured, we really looked into the temperature of the carcass over time, the extent of decomposition or breakdown, changes in the carbon-based nutrients, and changes in the soil or the nutrient leaching. Decomposition extent, I'll tell you a little bit more about it because that's one of the things we struggled assessing that, and I'll tell you our approach and look forward to your feedback. The last 2 points, we assessed it only at the 12 months point. So not at the 3 or the 6. It's only at the very end of the study, at the 12 months, that we actually dug up the carbon under the animals and then extracted soil cores up to 4 feet below the burial. For the decomposition scoring, we looked for kind of almost a consistency or a rubric for a way to arrive at a rank, sort of 1 to 5. Again, that's not a continuous scale, so you have to assign one of those 5 digits, 1 being the most intact or least degraded, and 5 is the most broken down. And we did not pioneer that this is from work on equine decomposition in Texas. So we use that as our rubric, and one of the things we've started to do is ask a lot of the help and the support. Many of them are composting SMEs, but also crew present and to support the project. What we instructed them is to tell them how the score to be interpreted and ask each individual to rank individually and not consult with one another. Essentially, and this is on the right is the scoring sheet that we've asked them to fill. So that was carried out at the 3, 6, and 12 months as well. So this is a site, what it looks like after essentially a week from the trial started, and kind of putting some seed on top to kind of Um, Jumping onto the results, we started to find some wildlife activity. There was no gray wolves, but we saw some um, bobcats, deer, dogs from the station. We saw some birds and predators essentially digging, especially the first 14 days right after the burial. It was very a strong attractant to where they actually have to come back and put some soil to make sure it's inaccessible to the wildlife of the site. So that was the kind of the one of the non-quantitative ways of looking at what we, sorry. Um, A little bit about the site temperature. Again, this is the coastal part of the North Carolina, so air temperatures and soil temperature, we were in July, so it was fairly warm, and the lowest temperatures we got were above 5 degrees Celsius. So it was, and you get to see soil and air temperature. Something I've neglected to mention, this station also has a weather station, part of a state network of climate monitoring. So it actually tracks soil and air as well soil moisture. So, and we've used that as well to help us with our analysis. Um, on the temperature side, we started to see slight increases in temperature compared to the site soil temperature with no carbon. And consistently, there were higher temperatures observed when you macerate the carcass, which makes sense. We are essentially introducing more carbon and available viscera for the breakdown. The only exception to that was using the mulch. We did not see a higher temperature with the ground carcass. The second point, I wanted to show you some of the visuals first for the decomp. Again, that's the 3 months, and I picked just using a hay as the material under the carcass, left to the carcass intact right after it's being macerated. We made sure that we dug through to make sure any residue of an animal is visible to the scorers or the individuals on the site. So it's an objective ranking. This is after 6 months. You can still visible signs of fats, rendering still, and tissue. It wasn't until the 12 months point that you started to see that disappearing. When we started to collate the numbers from the scoring the individuals, what you're seeing in the blue represents the whole carcass burial plots, the distribution of their scoring. So on the left is score of 1, 2, 3, and 5 at the very right. So most of the whole carcasses at the 3 months period were ranked on average at score of 2, fairly undegraded. Most of the ground were already very close to being decomposed. And you see a gradual shift rightwards for the whole carcass as you step forward in time, with that kind of quantitative way of scoring it. Again, this figure is trying to kind of aggregate everything by all of the variables, which is noisy. So I'll save you the time. And this is really the factors in orders of importance. It's the state of the carcass that was the most important factor statistically, then the age of the burial plot, finally the carbon. When we started to look at the carbon impacts, we found that the wheat straw was the best in terms of the degradation. Hay was actually surprisingly led to a better breakdown than corn stover and the hardwood mulch. A little bit on the site conditions, we actually got quite a bit of rain. So this is 200 millimeters, the maximum 7 inches and change. So this is the rainfall month to month during the year of the trial. When we tried to do the net between the rainfall and evaporation to get the sense of are we storing, are we drying the soil, we got that for most of the time we're actually in a wet conditions. The soil were quite wet, and the soil moisture content from the probes at the research station showed these moisture contents roughly between 20% and 35% soil volumetric moisture, which we expect, this is our field capacity, which means everything around that or higher, this is essentially saturated soils during the trial. So that had a great impact on what we saw. When we looked at the carbon again, and I'll give you a brief look in here. So on the left-hand side, that scale is showing the relative change in the phosphorus at the end compared at the beginning. The higher that bar, the higher the increase in the phosphorus. So you can see consistently that the ground carcass led to higher phosphorus in the carbon below it, which also makes rational sense that the nutrients were more available to translocate from the carcass to the carbon. The most we found in the hardwood mulch samples and the ground wheat straw. This is what we saw in the soil cores below the site. So this is ammonium. So to orient you, on the left-hand side, this is depth. So as you're moving downwards, you're actually going deeper in the soils. They are not actually to scale. So the first two rows is the first 6 inch, followed by the second 6 inches. But then after that, it's a foot by foot. Okay? And the very right-hand side where the arrow is, this is a background soil that has no carcass or mortality on top of it. So that gives you an idea that most we saw nutrients that bypassed the carbon and started to translocate below that, leaching. A lot of ammonium and higher concentrations. Typically with soils, we expect ammonium to nitrify and then hopefully denitrify, but we still, again, because of the saturation we observed at the site, that made sense that we saw much higher ammonium than what we saw on the site in terms of nitrates. Again, the nitrates was actually, we saw much lower with the exception of a hotspot in the samples. Most of them were below 20 parts per million, which is still elevated. So kind of to wrap up, as you can see, a lot of these factors interplay, the weather and the soil conditions in the site. We saw that the temperature had a strong dependency on the state of the carcass. We also noticed that the composition rate has a big dependency on the carbon type, the carcass, and how long it has been buried. We saw nutrients moving, leaching below into the soil. Again, whether it is reduced or nitrified, nitrogen in particular depended on the drainage and the rainfall at that site. I want to thank a lot of the good work and the support from the Pork Checkoff and the National Pork Board, as well as the station team, Smithfield Foods, for supporting the mortality. It's a big team to get that work done, and we're still analyzing the data, so hopeful to share the manuscript fairly soon. Thank you.